It's my distinct pleasure to welcome Alfredo to present today's seminar. Alfredo is a T32 Fellow on our basic science research in gastroenterology, T32 here. He is mentored by Manuel Kleiner, and he's at NC State University. A little bit about Alfredo. He graduated from Notre Dame in 2011 and completed his PhD at the University of Tennessee in Knoxville in 2020. I was really interested to learn that he's actually serving as a young alumnus on the Science Advisory Council at Notre Dame. Curious to know what that is.
Yeah, I mean, they have a council that they invite. It's mostly older people who are leaders in science, and they tell us about what the department, the College of Science is doing to sort of make Notre Dame a leader in that area.
So you're shaping science policy at Notre Dame. That's terrific.
Yeah.
Well, today we're really excited to hear about your talk. Dietary protein source strongly impacts gut microbiota composition and function. Take it away.
Alrighty.
Oh, and sorry, housekeeping. Please hold your questions to the end. Feel free to put them in the chat and we'll go over them at the end. Okay?
Alrighty. Hello, everyone. I am excited to talk to you today about the work I've been doing in my postdoc with Dr. Manuel Kleiner. The project is entitled Dietary Protein Source Strongly Impacts Gut Microbiota Composition and Function. In my talk today, I'm going to introduce the impact of diet on the gut microbiota. I'm going to talk about the experimental design of how we're looking at how protein in particular affects the gut microbiota. And then I'm going to show the effects of dietary protein on the gut microbiota composition. I'm going to explore the proteome of species responding to dietary protein. And then I'm going to show the effect of dietary protein on amino acid degradation. And last, I'm going to show you a very little bit about data of the effect of dietary protein on the host proteome. And so when I think about the gut microbiota, I'm really thinking about the microbiome that's in the colon, because even though there's a microbiome in every part of the digestive tract, in the gut, there's the largest population of the microbiome. It's an order of magnitude more microbes than any other part of the body. And in there, the microbes are playing a really important role in the function of the digestive system, where the microbes are engaging in the metabolism of the different metabolites to make it to the colon. And this metabolism has a very direct effect on the health of the host. And so in the gut, there's these microbes, and they have genes that express proteins. And these proteins are able to catalyze reactions that essentially process the different metabolites that can be found in the gut. And some of these nutrients come from the diet. And so that's a major part of the nutrient flow that's available to the microbiome. But the host also provides nutrients that the microbiome can access. And so the composition of the microbiome and the proteins that it's expressing together sort of manage the metabolite pool in the colon. And based on how this metabolism is going, we can have a strong, healthy mucosal layer that's sort of separating the microbiome from the host. The microbiome is feeding the host and helping the host sort of make best use of its nutrients. But in some cases, if the diet is not right, or if something's wrong with the composition of the microbiome, this function can look very different. And metabolites are generated that are toxic to the host. We see a degradation of the mucosal layer, and this can lead to sort of a compromise of the epithelial layer that is sort of a signature of disease in the gut. And so diet plays a major role in the nutrients that are available to the gut microbiome to sort of drive this metabolism. And here I'm just showing you a few examples of why, as you change diet, the nutrients available to the microbiome change. And so here is a figure from a Cell paper published a few years ago, sort of shows the composition of polysaccharides derived from citrus protein, P fiber, and you can see
Protein mutant controls. The next one. Yeah, that one.
Sorry, I just want them to be able to see.
Okay, well, I apologize for the technical issues. So like I was saying, depending on what you're eating in the diet, a very different profile of polysaccharides will be available to the microbiome to be eaten. And, you can begin to imagine how this will immediately alter which microbes are able to survive in the gut and the types of metabolism and products that will ultimately result. And it's not just fiber that is providing these polysaccharides to the gut. Different proteins that we eat in our diet, for example, eggs, the proteins in there are also glycosylated, and they have very specific structures which only certain microbes can access. And those sugars can also be different than, say, another fiber or another protein. And even the next level, the host also provides sugars to the microbiome, particularly mucin, which makes the major portion of the mucosal layer, has very distinct structures of different sugars that the microbiome can access. And so the microbiome, so the microbiome has evolved different mechanisms to access these polysaccharides. And different microbes have different mechanisms and have the ability to degrade different sort of structures. And so the main way that they degrade them is with glycoside hydrolases, which are able to sort of cleave sugars off of a complex polysaccharide. And different glycosyl hydrolases are able to break basically different bonds. And one of the systems that is most interesting or most well characterized for accessing these sugars is these so-called polysaccharide utilizing loci, which are present in the phylum Bacteroidetes or Bacteroidota, depending on which taxonomic dogma you are following at this moment. This system is very interesting because basically all the genes needed to degrade a specific polysaccharide complex are present next to each other in a gene neighborhood. And so the hallmark of these systems is a SUST-like proteins, which are basically the transporter, which imports the sugar. And then there will be different glycosyl hydrolases in the system that will be responsible for degrading different types of bonds. Like in this figure here, the GH18 breaks this bond between two acetylglucosamines. The remaining polysaccharide is then imported into the cell, and the different mannosadases in the gene neighborhood are able to cleave break down the rest of the polysaccharide into individual nanosugars. And these organisms from this phylum have basically many different polysaccharide utilizing loci to degrade polysaccharides, different polysaccharides. But right now I've been only talking to you about sugars, but purified dietary proteins can also have very different glycan and amino acid content. Different sources of protein will have different amounts of glycosylations attached to them. So this data is from some purified protein samples we sent to Eurofins for analysis, and we got back different values of amount of sort of glycan that was attached to these proteins. And we also analyzed these purified proteins for amino acid content, and you can see that there are different proportions of the different amino acids in these proteins. And so one can also imagine that different proteins provide different nutrients to the gut, which would have an impact on the microbiota. With the different proportions of amino acids, one also has to think of themselves that when amino acids are being degraded in the gut, different amino acids have different metabolites that are produced. And depending on the microbes available, they're also processed in different ways. So here's like two different pathways of L-glutamate, right here and right here. And you can see that depending on the organisms available and the enzymes available, you're going to get a very different metabolite pool. And so all this serves to just kind of me present to you that what we eat provides very different nutrients to the gut microbiota, and we should not be surprised when what we eat has a massive impact on the gut microbiota. And this can be shown by the literature. So in this study, humans were basically fed a diet where fiber was taken away. Basically, they switched from just a normal diet to an animal-based diet, and that animal-based diet had basically the signatures of low fiber, high protein, and the composition of the gut microbiota changed in just a couple of days. And then when they went back to their original diet, it recovered. And other literature has shown that this kind of dietary change can have a detrimental effect on the host. So in notobiotic mice, in this study, they fed them a low fiber diet. And as a result of this, they observed a degradation of the mucosal layer. These mice became more susceptible to infection, and bacteria that are involved in the consumption of mucin increased in abundance, and their mucin degrading functions were increased. And I've seen this kind of effect in my own research. In my dissertation, I looked at the metaproteome of mice that were lean, eating a standard chow, genetically obese, but also eating a standard chow, and fed a high-fat diet that was 60% fat, but also had very low fiber and a purified protein source. And in there, we see an increase, and in that study, we saw an increase of enzymes associated with the degradation of sugar, with the cleaving of sugars that are typically associated with mucin. So we see this effect in my own research as well. And so at this point, you're thinking fiber, 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 but you said this talk was about protein. And the thing is that in all of these studies, while the statements are about fiber, they also are seeing a change in the source of protein. And in the case of the standard high-fat diet that a lot of different studies have used, and the notobiotic study, the protein source was switched to a purified casein. And understanding how protein affects the gut microbiota is important because several sort of large cohort studies have shown that diets high in animal protein have increased mortality relative to diets that are not high in animal protein, and that this effect is not seen when people eat a diet that's high in plant protein. And in a recent one of these studies, they actually found that specific animal proteins had increases in mortality that were greater than others. For example, egg had the highest increase in mortality, followed by red meat, 24% and 21% increases in mortality, relative to a high plant protein diet. And so kind of based on that, our question is, okay, so how does source of dietary protein alter the composition and function of the gut microbiota? And in the images here, I'm sort of showing different places where our protein can come from. And so we designed an experiment to sort of go after that. And so I'll describe the experimental design. And so in collaboration with a former master's student from our lab, Ali Bartlett, and Casey Thiriot's lab, we designed diets that were sort of defined and where all the nutrients, at the best of our ability, were maintained the same. And then we had a purified source of protein that represented 20% of the diet by weight. And so then we designed several different versions of this diet, which each had a different source of protein: soy, casein, rice, yeast, pea, and egg. And then we also had a chicken bone diet, which we didn't get to use because it had too strong of an effect on the mice, and so we had to abort the study at that point, bring them back to a standard chow, and then at the end we had a sort of control diet where we fed them half the mice the original soy diet and half of the mice fed the original casein diet. And so the way this study is designed is that each mouse is fed one of these diets for 1 week, and at the end of the week, we collect a sample. And the sample is subfecal sample. And at the end of the week, we transition to the next diet, and sort of we continued each week. And then in some cases, we increased the amount of protein, but went back to an original source of protein. So in this case, we increased to 40% soy. And in this case, we increased to 40% casein. And at the end, we have our control diets. And so then to sort of explore the gut microbiota, we did it in a bit of a different way than you would typically see in most microbiome studies. We took the fecal samples and then we analyzed them using what I like to call an integrated metagenomics, metaproteomics workflow. And so basically what we did is we took the fecal samples and we extracted DNA from them. We submitted them to shotgun sequencing, and we did sort of an advanced bioinformatic process to taxonomically classify the results of the sequencing and build a list of sort of a catalog of proteins that could potentially be in the samples. And then to these lists of proteins from the metagenome, we also added the host proteome, and then we sort of predicted the types of dietary proteins we thought might be in the sample as well. So this is sort of our foundation of our study. And then we extracted protein from the fecal samples, digested them into peptides, submitted to a mass spectrometry system coupled to liquid chromatography, and employed a method called tandem mass spectrometry, which basically takes the peptides, fragments them into a defined pattern, which using an algorithm, you can match to the protein database to identify peptides, which can then be used to infer and quantify proteins. And so when you do this type of analysis, you're left with a table that has a list of proteins. Hopefully they're assigned taxon, and then a series of abundances. And from this data, you can actually do a lot. So you can take this data and aggregate it by taxon and do your typical taxonomic compositional analysis. You can look at the individual proteins and try to find out if there are specific proteins that are different between conditions. You can also aggregate these proteins into functional categories and sort of do general metabolic analyses. And then you can also, which is not kind of shown here, is you can segregate proteins belonging to specific taxa and do within proteomic analysis. And since we added the host proteome and a dietary proteome to our database, we can also detect those proteins. And so using this data, and so to clarify, there is no 16S data in the study. There's no 16S data in the study. I get that question a lot, so I'm just making sure we know that at the front end. And so using this data, I was able to do a general compositional analysis like you would do with 16S data, but we're using protein abundances attached to our taxa. And so we started with a sort of basic alpha diversity analysis using the Shannon diversity index, which kind of gives us a measure of the complexity of the microbiome under these different diets. And very quickly, we see that the microbiome collapses under the yeast and the egg diets, but sort of recovers when we go back to the controls. And we also see that there's a bit of a dip after the yeast. So there's that effect is retained a little bit, even though there's a little bit of recovery. Sort of an interesting measure that we're playing with that's available to us in the metaproteome, because we have the microbial proteome and the host proteome together, is we can do sort of a microbe to host ratio analysis where we divide all the microbial proteins by all the host proteins. And we're sort of able to create a sort of a measure of increase of bacterial load. And we see that the bacterial load is, even though the diversity is dropping, the bacterial load is increasing in the yeast protein diet. And then finally, we're able to do sort of your typical beta diversity analysis, how dissimilar or similar these microbiomes are to each other in composition. And so this is Bray-Curtis dissimilarity, where high numbers mean that the microbiomes are more different from each other, and low values mean that the microbiomes are more similar to each other. And what's plotted here is basically the comparison of all the 20% soy protein diets to all the other diets in teal, and 20% casein against all the other diets in red. And what we see is that the microbiome is most similar to to the same dietary protein. So if we trace the 20% soy, we see it drop when it's comparing against itself. Again, when we increase the 40% soy, and then again when we return to the soy control. And we see the same effect with casein. And so what we're seeing here is that type of protein has a greater effect on the composition of the gut microbiota than amount of protein. But this effect is not the whole story. Something that I neglected to mention, but may have been visible in the diagram, is that our mice came in two groups. They're half were male and half were female, and then each were in two cages. And the male and the female mice are from different birth rooms. So they have a different starting gut microbiota. And when you do principal component analysis of the species, you can actually see this separation. And so there's no cage effect. If they're from the same birth room, they have the same similar microbial composition, but the two groups have a clear separation in microbial composition. Despite that, we can still see the separation of the diet based on the yeast and the egg diets down here. And if we focus in on break the groups apart, we can see that there's sort of a casein group, a soy group, a standard chow group, and a yeast and egg group. And this is also visible in the male mice, but the separation is not as distinct as in the female mice. So, you know, from this effect, we can say something about how starting microbiota, has an effect on this, but we can't say whether it's male or female or the birth. And so then, you know, driving this forward and trying to piece apart what we're seeing as the effect of dietary protein on the gut microbiota. So group 1, which is the females, and group 2, which is the males. If we roll our taxonomic information up to the family level, we can see a rolling change in the most abundant family as we switch from dietary protein source to dietary protein source. We can see recoveries when we go back to an original dietary protein source. So here we can see that Lactospiraceae increases in abundance in the soy diets, here and here. Here and here, and then here and here again. And then we can say that Bacteroideaceae is sort of the signature of yeast and egg. So it increases here and here, and here and here, but that the different starting gut microbiotas have different responses. So here we see Akkermansiaceae increasing in the females, and Moribaculaceae increasing in the males. And we can sort of go with this sort of data, we can go down to the species level, and so we can actually see the individual species that are changing. And sort of to reduce the complexity of the data, I reduced the data set to any species that was 5% of the proteome in any given one sample. And so these 36 species, each were very abundant species at some point. And we can see here that the microbiomes do segregate by, you know, group 1 and group 2, so the males and the females, but that the yeast and the eggs diets separate out into their own group, and it's driven by a massive increase in Bacteroides micron. And we can also see that in egg, there's sort of different secondary species that are increasing in abundance. We can see here Akkermansia muscipula for the females, and a as of yet unnamed species, but from the Actophaceae bacterium family. And then we can see a novel genus, a genus that's been recently characterized called Paramuribaculum in the males. And we also can see that the microbiome is collapsing in general in these two yeast and egg diets. And we can see that there's a very distinct overall population between the males and the females and the males. And so kind of the question we wanted to ask ourselves is, what is happening in the proteome? And can we begin to understand this yeast and egg effect? And so we brought the analysis down to the microbial protein level and sort of did a similar sort of hierarchical clustering analysis. And once again, we see a separation of the yeast and the egg diets from the rest. And when we dig into these proteins, we see, we lost the names here, I apologize for that. But what I'm trying to show here is that we see Bacteroides micron play a large role in these heats, and we see a few different secondary species in the male and the female egg diet. So we wanted to learn a little bit more about what was going on with Bacteroides micron. And so Bacteroides micron is sort of the major driver of what's separating yeast and egg from the remaining diets, because it's increasing massively in abundance. B theta accounts for up to 80% of the protein biomass in the yeast diet and up to 50% of the protein biomass in the egg. And at this point, you might think, okay, well, Bacteroides micron is likely doing something similar in the yeast and egg diet. But when we looked at the proteome of Bacteroides micron, so what we did here is we extracted the proteome from the metaproteome and renormalized it so we can analyze Bacteroides proteome within the microbiome directly. And out of 1,400 proteins we detected in the study, 592 of them were significantly different, and 30% of these expressed proteins would belong to what I just described earlier, polysaccharide utilizing loci. And so what we're seeing is that Bacteroides micron is expressing different polysaccharide utilizing loci between the yeast and the egg diet. So it's doing something different to go after these yeast and egg diets, and it's responding in a different way. And what we can see is that basically these gene neighborhoods are being expressed together in one of the diets. So for example, here's PUL71, which has 3 mannosidases at the end and a sus-C, sus-D like protein at the beginning. And we can see that it's expressed in, all the proteins are expressed in yeast and not in egg. And we actually detected several polysaccharide utilizing loci with similar signatures, so mannosidases. There's a couple different other types of glycosyl hydrolases, but mannosidase was really the key signature. And that makes sense because the yeast cell wall consists of about 50% mannoprotein. And so Bacteroides micron is likely using these polysaccharide utilizing loci to degrade these mannoprotein and use those mannoses for its own metabolism. And some RNA-seq data that's been done on some of these PULs indicates that at least some of these PULs have previously been shown to be associated with degradation of mannan. And so we're seeing sort of a mannan to mannose metabolism that Bacteroides micron is doing due to the yeast diet. And on the egg side, we got a different set of polysaccharide utilizing loci. This one, PUL16 from the CASI database, has GH18s, which cleave acetylglucosamine bonds. And you once again see sort of the sus-C, sus-D, and all the proteins expressed in the egg diet, but not in the yeast diet, or very low expression in the yeast diet. And there's a whole nother series of PULs with GH18s and other acetylglucosamine degrading glycosyl hydrolases attached to them. This one's a little bit interesting because while it has mannosidases like the yeast PULs, it's actually a PUL that's been associated with the degradation of high mannose mammalian proteins. So it still fits with the story, but it also has mannose degradation, mannan degradation enzymes in there. And one of the things that was interesting about this is if we recall the components of mucin, acetylglucosamine is a major component, and previous study have linked some of these PULs to the degradation of mucin. So we're starting to see a link between mucin degradation and egg white degradation. And if we look at the structure of proteins from egg, this sort of story starts to make sense because they are covered in acetylglucosamines and mannoses, and different studies have sort of shown the profile that they may have. So at this point, what we wanted to do was sort of prove that Bacteroides micron was actually growing on these dietary proteins, and that it was employing these sort of proteins to do it. And so we collaborated with one of the students in Casey Theriot's lab, Sam McMillan, to attempt to create medias of these dietary proteins and grow Bacteroides in these medians with these dietary proteins as the sole source of carbon. And so we had a minimal media with no carbon source, a glucose minimal media as a control. Then we had protein controls, casein and soy. And then we had our mucin control. And then we have our yeast and the egg. And we basically put Bacteroides into these minimal medias and at a sort of starting CFU that you see here. And we see that Bacteroides did indeed grow in yeast, egg, soy, and mucin, and that it grew best in yeast. And there's no significant difference between egg and mucin. And it grew a little bit in soy, but not as much. And so then we employed traditional single organism proteomics on this data set. And this is hot off the press, just a week old. So we haven't dug into it too much, but we can already see it clustering in a way we would expect, where glucose has its own proteome that's very different from the dietary proteins, and mucin and egg are clustered separately but together. Sort of going with this study, B theta is not the only mucin degrader present and growing in the egg white diet. When we look at the sort of representation of the metaproteome of the egg white diet in the males and the females, we see species such as Akkermansia muscipula and Paramoribaculum, which the literature has been indicating for many years are bacteria that grow on mucin. And when we look at the proteins of these different species, we see glycolysis, we see fucose, acetylglucosamine, and sialic acid metabolism. We see amino acid degrading pathways. So we see all the things that are sort of indicative of the metabolism associated with mucin degrading, but in the case of an egg diet. Other literature has sort of indicated that there may be a link between the egg diet and bacteria that can grow on mucin. So for example, Akkermansia muscipula in this study was shown to grow on ovomucin, which is a component of egg white, but ovomucin is a very small part of the proteome of egg white. So this is not yet that established, but it sort of indicates Akkermansia may have the ability to grow on egg white. Here in this table, I'm showing you the dietary proteins we detected in the fecal material that are most abundant in our fecal samples. So, at the top we have of albumin, and down here we see ovomucin, a very low abundance, but many of these proteins are N-linked glycosylated, although it's not yet so clear what those look like. So there's a lot of potential here for sugar that can be used to grow in a very similar way to mucin metabolism. And so future work will be sort of exploring that side. The carbohydrate degradation effect of dietary protein sort of rolls up to the whole microbiome. So here I have the composite abundance of glycosyl hydrolases of my proteins as annotated by the CASI database. And so you see massive increase in total glycosyl hydrolases in yeast and egg. And then when we normalize across all, we can see segregations into yeast, egg, standard chow, soy. And then there are some casein groups, but those are not as defined. And we can see that different glycosyl hydrolases like these different diets. And so from here, we can say that dietary protein affects the composition of the gut microbiota through the glycosylations attached to them. But, you still kind of want to know what the amino acid backbone is doing to these gut microbiota. So I went around to come up with a way to sort of hunt for amino acid degrading enzymes and to see what was going on in that sphere. And so to do this, I used the Metapsych database, and I basically went pathway by pathway, selecting the first enzyme in the pathway. I then looked at the annotations of my proteome and aggregated the protein abundances of each that was annotated with one of these enzymes, and basically created sort of like an amino acid degrading abundance enzyme table. And when we aggregate this all together, we see that very specific diets have sort of an increase of general. So like, for example, we have rice here, we have egg here, and interestingly, we also have 40% casein. And when we look at dietary proteins in the fecal material, this actually makes a little bit of sense. So we detect the most dietary proteins in our fecal material in the rice and egg diets. You know, this is a little confusing here because we don't see a large amount of casein proteins in our diets, but casein doesn't come alone. It has a hitchhiker. L-lactis is present in large amounts in all sort of casein protein that you feed to mice. And we detect it in very large amounts in our fecal samples. So this is not a member of the microbiota, it's actually a dietary protein, and it's present in casein. And so from here, you know, it's the vision is not perfectly clear, but we can start to see that amino acid degrading enzymes increase when dietary proteins are detected in the fecal sample. So they make it all the way to cases. And we can also, when we look at the individual amino acid degrading enzymes, we can see that specific amino acid degrading enzymes tend to spike across different dietary proteins. And so the next step is sort of to dig into that a little. And so the last thing I wanted to show you today is that we haven't spent too much time looking at the host proteome, but our metaproteome does come with a host proteome. And you know, we wanted to see if there were some significant effects in there, because it's really an aspect of metaproteomes that's underutilized. And so what I did is I wanted to look for, okay, what is the host proteome signature of our yeast diet? And to do this, I basically compared significance between, I extracted the host proteome and renormalized it. And then I sort of compared significance between yeast and all the other diets, and focused in on just the proteins that were significantly increased in yeast versus all the other diets. And we see these reg3 beta and reg3 gamma proteins, which are lectins associated with antimicrobial function. So they basically kill bacteria, which is interesting because the complexity of the microbiome is dropping, and Bacteroides is blooming, and reg3 gamma doesn't affect Bacteroides So there's something interesting going on here, but I don't want to oversell it. Another thing that's interesting besides adenosine deaminase, which has been associated with a lot of inflammatory diseases in the past. On the egg side, I did a similar analysis, and we identified a lot of different proteases increase in egg, which makes sense because there's more egg protein making it to the colon and into the fecal sample than the other diets. And then another interesting thing is that we actually saw IgA be significantly increased in egg and yeast compared to the other diets. And so with that, you know, I would like to say that dietary protein can have a large impact on the gut microbiota's composition. Gut microbiota feed on dietary protein by degrading the glycosylations. Enzymes typically associated with mucin degradation could also be involved in the degradation of dietary glycoproteins. And amino acid degradation appears to be tied to the digestibility of the dietary protein. And so the impact of this is that source of dietary protein should also be considered when tailoring diets to gut microbiota. Taxa abundance alone is not a measure of function. With the proteome, you can actually see what those taxa are doing. And even if a taxa is abundant, it could be doing something different under different conditions. And then the last thing is that protein glycosylations are an important nutrient for the microbiota. With that, I would like to thank Manuel Kleiner and his lab for all of their support. In particular, Ali Bartlett, who played a major role in designing this experiment, and then Alyssa Ehrenhoff, Molly Van Hoy, and Aisha Awan, which are helping me with a lot of the follow-up assays of the study. I also like to thank Casey Tiriot's lab, Casey Tiriot for her mentorship, but also her lab for doing the mouse experiments, and Sam McMillan in her lab for helping us with culturing of the Bacteroides. And then on the side of my CGIBD mentorship committee, Lawrence David and Balfour Sartor have provided a lot of insight into our interpretation of this data. And so thank you for that. And these are my sources of funding. If anyone out there is looking for a postdoc in a similar project, you can let us know because we have funding for that. This is a picture of our lab. See how happy they are. With that, I will open up for questions.
So I was kind of like taken with this, something you just said about understanding more about like what the bacteria are doing. You look a lot at like bacterial metabolism and everything, but can you look at other aspects of the microbial behavior, like their reproduction and things like, you know, like whether they're countering some of these increased inflammatory responses from the host, or kind of more at that intersection of like host and microbial kind of back and forth?
All of that is possible. You know, one of the reasons that I focus on the metabolism for two reasons. One is very practical. Metabolism as a function, because the protein do specific reactions, is very straightforward to sort of tease apart function in a very specific way. And so when I think of microbial function and proteomes, building a pathway is much more straightforward when you're looking at metabolism. Answering your question about other functionalities, a perfect example that I didn't show here is that ribosomal proteins are increased in the yeast diet, which makes sense because Bacteroides grows faster in yeast than it does in egg. And, you know, it's blooming to such a degree that it's sort of breaking the balance of the ratio between the microbiome and the host. And so, you know, there's that aspect that we could definitely talk more about. The last bit is actually practical from the perspective of what the microbiome does in relation to the host. So a lot of the functionality that the host responds to is metabolic. You know, the most classic examples of the microbiome modulating the immune system are the short chain fatty acids that are being produced from these metabolic processes, and the toxins that are produced from degrading amino acids and all sorts of things. You know, the feedback between Akkermansia and the host is such that if it's not there, the mucosal layer looks bad. If it's there, the metabolism of the mucosal layer spurs the production of more mucus. But then if it's too abundant, if there's not enough dietary nutrients, then, you know, it goes bad again. And so that dynamic there is why metabolism is such a focus of my functional research.
Heather?
I'm curious about why you had to stop the experiment with the chicken. Could you?
Yeah, actually, that was my question too. What happened with the chicken?
Oh, the mice got very sick.
How
But
sick?
did they not eat, or did it collapse the microbiome? Did you look into it at all?
We weren't even able to get a sample. They sort of just got very, very sick. I mean, you know, I didn't do the mouse experiment, so I can't provide too much detail on like how the mice were sick. I just know that, you know, the technician responsible was like, the mice are too sick, we need to stop. And fortunately, we didn't lose too many. Well, we didn't lose very many at all. There was one or two mice that died along the way, but I'm not sure that any died in that moment. And so then we moved them to standard chow, they recovered, and we were able to do the control experiment.
That was bone meal, right? It's chicken bone meal?
Yeah.
Yeah.
A comment on that, Jeremy anecdotally told me a story about a very similar like animal protein diet that they put their mice on, and colitis came to be within 3 days. And they also had to stop the study, so maybe talking to him would give you some insight. But my question, I have two questions. So the first is, you started with soy, you ended with soy, or you started with casein, you ended with casein. How similar were those proteomes?
Oh, well, I think I showed that.
Do you have any differences or?
I mean, there's like, sort of like stochastic differences that just kind of happen. But if you look at the, index of the similarity, it does come back down.
I guess I'm interested in the proteome specifically rather than the bacteria that are there.
Yeah, the proteome mirrors the bacteria.
Okay.
So like abundant bacteria, we get way more proteins.
Okay.
That's why the proteomics here is like possible in Bacteroides microns because it's so abundant. We're getting lots of Bacteroides proteins that we can do a proteome.
I guess, so maybe that answers my second question. So I know that there's that really famous Nature paper showing that when you do 16S sequencing and you do phylogeny, but then when you do the metabolome, it clusters very differently to what the 16S shows. So like you have all your phyla nicely, distributed in your 16S, and then we look at metabolome, like all of a sudden the bacteria, these are no longer together, like they're all interspersed and the firmicutes are all over the place. So based on the metabolites the bacteria are producing, when you cluster by metabolite production, it looks very different than when you cluster by 16S taxonomy. And I guess I'm wondering here, when you cluster by the proteins being produced, do they still cluster like you would by taxa, or do they look very different when you're clustering by the proteome rather than the bacteria? Does that make sense?
Yes. So in general, the proteome will mirror abundant taxa. But, like I showed in different instances throughout the talk, you can aggregate function across taxa and show an overall functional effect. But, actually, in this paper, I don't have the other figures surrounding it, but in this paper, which I published very recently, we see a separation between the genetically obese mice and the diet-induced obese mice in taxa space. But when we look at function, sort of diet is having an effect, but the genetic portion is not, the effect is not a strong function.
I guess I'm wondering, like, do firmicutes produce very similar proteins in all firmicutes, or?
Well, I mean, proteins are totally distinguishable between species. Like, they're not the same.
Okay.
So functionality could be similar if the proteins are homologs, but even the same species could have very different functions. I mean, within E. coli, for example, the proteome can range from 2,000 genes to 7,000, depending on the strain.
Okay.
There can be extreme heterogeneity in the functionality available for different taxa. And I would be suspicious of any paper that claims a metabolite belongs to a specific taxa.
I have two questions. First, thank you for that very informative one. Going back to the mice not tolerating the chicken bone. I know that in broiler chicken, if you feed them like a high animal and bone diet, they get a lot of Clostridium perfringens, and they have trophic enteritis, and that leads to a lot of mortality. So I'm wondering if that's something that could happen also with mice, and if that happened, could that affect the follow-up weeks because of the recovery of the effects for the gastrointestinal tract? And in one of your figures, you mentioned that I can't quite not remember, but you mentioned that there was a dip and then it quite did not recover back. So I'm wondering if it could be the post chicken bone. Yes.
Yeah, so that's, chicken bone occurred here. So the collapse of the microbiome is due to yeast, but the mice are still like good enough to continue the study. And then the microbiome recovers a little bit, but not, it's not all the way to the top. But by the end of the study, we're sort of back.
Yeah. So
And this is after sort of the chicken bone. This here is after the chicken bone.
So you're not worried about like any like physical damage happening to the gastrointestinal tract because of that diet?
Oh, I think that's very hard to say. You know, I think we would not repeat that. But I think that there is definitely a lot to learn from exploring more realistic protein sources. And there's also a whole aspect that I haven't talked about here is that, you know, they were on a very defined fiber source. But if we alter sort of the other nutrient, you know, here the point was how does source of protein affect the gut microbiome if everything is equal? But I think that there will be more dynamics when we change other nutrients and other proteins together that still need to be.
And my second question is, I might miss it, but what was the reason for you to pick a higher protein percentage at the middle of the experiment for the soy and the like, why did you switch from 20 to 40?
Oh, I think the idea was there just to see if increasing diet, you know, I think when we started, since most of the studies kind of look at increasing protein, we were sort of hoping that I think at the beginning, that was one of the ideas of where the greatest effect would be, even though it turned out not to be the case. Yeah, I think actually that 20% is already a high amount of protein. And so you might see a much stronger effect if you dropped protein. In fact, you know, one of the themes that I'm seeing in a lot of these studies is that, you know, once it gets to a certain baseline, more is not going to change it. It's really like when you take a nutrient and you change a nutrient that we see big effects.
Same question.
Yeah.
Okay, so for questions on Zoom, have you tested combinatorial effects of your protein sources on microbial diversity and the proteome, in particular a diet of yeast and soy? Would the profiles be intermediate, or do you think one protein source would dominate?
We have not looked at that. So, you know, anything I would say would be speculation, but I would imagine that if there was a protein source, the specific bacteria was better at getting. So in the case of yeast, Bacteroides seems to be very good at getting Bacteroides at the expense, getting it at the expense of everybody else. You know, maybe, you know, I don't know. That would be a great study.
Thanks for a terrific talk. Yeah, it's really great. Any other questions on Zoom, folks? Guess not. All right, everyone. Thanks so much for making the drive here.
My pleasure.
Great job.
Changes things up.
Thanks, Alfredo.